Part B of Chapter 5 on microbial metabolism, we're going to talk about fermentation. So in our overview, we discussed previously in Part A respiration, and we saw that if we start with one molecule of glucose, take it through glycolysis into the Krebs cycle, shunt it into the electron transport chain to harvest our energy from our NADHs and FADH2, we can get the most bang for our buck and get 38 ATP by aerobic cellular respiration. Fermentation, on the other hand, is going to be something that is a low energy yield, but it gives us a lot of nice fermentation products. So what is fermentation? We don't like to think of it this way, but fermentation is any spoilage of food by microorganisms. What most people think of if you say fermentation is probably going to be the process that produces our acidic dairy products or our alcoholic beverages. In reality, in the industry, these have to be ramped up to large scale. So any large scale microbial process with or without air to be able to generate these types of beverages and dairy products. In reality, a little bit more scientific, any energy release that is involving only the anaerobic conditions is still classified as fermentation. However, when we truly get to the true definition of fermentation, it's any metabolic process that releases energy from sugar or other organic molecules, does not require oxygen or the electron transport system, and uses an organic molecule as the final electron acceptor. It's almost like it can't get all the energy out of that organic molecule, it just kind of stops there as that organic molecule serving as the final electron acceptor, but it generates enough ATP to be able to keep things going. So there are lots of nice fermentation products. We start with our glucose molecule, go through glycolysis and convert that into our 3-carbon pyruvic acid, and then different organisms will use pyruvic acid on different pathways to make either acidic or alcoholic end products. So we see things like the Proponium bacterium. This is going to be useful in making Swiss cheese. So the holes in Swiss cheese comes from the carbon dioxide, and the propionic acid is what gives Swiss cheese that unique flavor. When we think of things that make lactic acid, those are the bulk of what we use for making our dairy and some of our soy sauces and various things like that. So these are going to be things like Aspergillus and Lactobacillus streptococcus. For alcoholic beverages, Saccharomyces, which is actually a fungi, a single-celled yeast, and is eukaryotic, but it grows pretty much like a bacteria in many ways. So it's very easy to grow, and we use that for making most of our alcoholic beverages. The bubbly comes from the CO2 that's being produced, and the ethanol that gives us the alcohol in things like our wines and beer. Things like Clostridium we use to make isopropanol, rubbing alcohol, and acetones. E. coli and acidobacters we can use to make acetic acid, and that can be used for making vinegar. So lots of things you're familiar with. So fermentation is, again, by definition, the release of energy from oxidation of those organic molecules. But the oxidation just doesn't go very far. It does not require oxygen, and we can't use the Krebs cycle or the electron transport chain, so we don't get a lot of bang for that molecule of glucose or whatever the organic molecule is that's coming in. An organic molecule serves as the final electron acceptor, and it only produces a small amount of ATP, only 1 or 2 ATP per starting molecule. So at max, for every molecule of glucose, it can make 2 ATP. The final electron acceptors are reduced to the fermentation end products that we saw in the previous slide in being useful products to help make our beverages and dairy and vinegars, etc. One of the things that we don't think about of the importance of fermentation is that it ensures a steady supply of the oxidized form of the coenzyme, the NAD positive. That's important to feed back into glycolysis so that metabolism doesn't come to a screeching halt. NADP is also recycled in this way, but we're not really talking about the anabolic processes here, so it's kind of just a sidebar at the moment. We're going to focus on the NAD positive again. So again, pyruvic acid is that point where different organisms then utilize the pyruvic acid differently. So again, the most useful things like Streptococcus, Lactobacillus, Bacillus, making a single lactic acid is very useful. Things like E. coli and Salmonella, they make useful products, ethanol, lactic acid, succinic acid, acetic acid, but you have to then purify to get the individual end product that you're interested in. So each of these kind of has their own uses. Again, the bulk of our alcoholic beverages are going to come from the yeast, the Saccharomyces. So fermentation, there's two basic pathways that are going to split off of the pyruvic acid once that is made from glucose in glycolysis. When we talk about the lactic acid fermentation pathway, there are some organisms that we just saw, things like Lactobacillus. It gets its name because it can only produce lactic acid. Those are referred to as homolactic fermenters. Things like E. coli and Salmonella that we saw, yeah, they make lactic acid, but they also make other compounds. So those are referred to as heterolactic fermenters. And again, those other end products may be useful. You just have to figure out how to purify them from the rest of the products that are being produced. So if we look at the lactic acid fermentation going down the left-hand side, again, we start with glucose and we take it through glycolysis as normal, producing our 2-ATP and our 2-NADH, and we get our 3-carbon pyruvic acid. If it goes into lactic acid fermentation, what we see here is now you're going to take the NADH, and that is going to give up those hydrogens so that you're now going to have the oxidized form of the NAD positive, and that is actually going to recycle back into glycolysis proper to allow glycolysis and metabolism to keep going. As those two hydrogens are released from NADH, those are used to turn pyruvic acid into lactic acid, and again, you can have other acidic products being made off of that pathway. The alcohol fermentation produces ethyl alcohol and carbon dioxide. So at the pyruvic acid level, after glycolysis, we see the first thing that's going to happen, kick out the bubbly, so you kick out the CO2, and this is going to leave you with a 2-carbon acetaldehyde. Acetaldehyde is then going to be turned into ethanol because, again, the reduced form, the NADH, is going to donate the hydrogens, and that is going to be used to convert the acetaldehyde into the ethanol. The 2 NAD positives, again, will return to glycolysis proper so that recycling ensures that glycolysis is going to keep going. Yeah, we're only going to get 2 ATP, but it's going to allow the cell to survive. And that's part of the reason why organisms that can only grow by fermentation are going to grow very slow. So again, some of the nice, useful, different types of fermentation products. So if we look at things in this list, sometimes we see some of our nice ethnic foods. So when we talk about things like sauerkraut, when you look at the starting material, cabbage would go bad very quickly. If you ferment it to sauerkraut using Lactobacillus plantarum, this is going to allow that sauerkraut to be much longer lived. It's not going to rot, basically, as cabbage would. And that's why we see fermentation from very ancient times was utilized to help make products that were used for food consumption to have a longer shelf life, even in ancient times. We see things like our summer sausages. Meat would normally start to decay very quickly, but if you start to ferment with the pediococcus, you make things like summer sausage, which are these nice hard sausages that you see hanging in the market. So we see lots of interesting products being made. Once you have a good strain of Lactobacillus debrecii, the rye bread, those can become a strain that is highly prized because they give a certain flavor to the bread that you're making. So not only are these foods, but we also start to see that sometimes we can use things like that acetic acid and use things like the methanosarcini, which are going to help us to make fuels. So there's lots of diverse reactions that can be harvested for industrial purposes because of fermentation. And lest we forget, just a reminder, all the good things that we get from fermentation. Hopefully this isn't right before lunch or dinner, or if it is, maybe you'll go and have a nice fermentation product at this time. So if we compare ATP produced by various metabolic processes, we can look at the amount of ATP and the processes for aerobic respiration compared to anaerobic respiration and compared to fermentation. So aerobic respiration, the growth conditions obviously are going to be aerobic, and the final electron acceptor is oxygen. It is going to be able to generate ATP by both substrate level and oxidative phosphorylation. And for a prokaryote, for every molecule of glucose that comes in, we get 38 ATP. For eukaryotes, again, it's a little bit less, somewhere less than 36, typically between 32 to 36 ATP. Anaerobic respiration has to occur under anaerobic growth conditions. The organisms that carry out anaerobic respiration, like Clostridium, they are actually killed in the presence of oxygen. They are going to have an inorganic final electron acceptor, but not oxygen. So things like the nitrates and sulfates and carbonates, similar to aerobic respiration, they also do substrate level and oxidative phosphorylation, but the amount of ATP that they could produce is always less than 38, which is aerobic respiration level, but more than 2 ATP fermentation level ATP production. So the amount of ATP produced is going to be variable depending on the specific organism. And this is because not all of Krebs cycle can be utilized, so therefore we don't get as much NADH and FADH, our foreign currency, to be able to generate as much ATP. Fermentation is the lowest energy yield. One of the things to remember is that the growth conditions can be aerobic or anaerobic, but this depends on the type of organism that you're dealing with. If you have an organism that is strictly fermentative, it can only carry out fermentation, it doesn't care whether the growth conditions are aerobic or anaerobic. That's all it can do. But if you have something that's a switch hitter that can go back and forth between aerobic respiration and fermentation, then in order to get those organisms to carry out fermentation, you do have to get all the oxygen out of the environment. Otherwise, it would want to carry out aerobic respiration because it would get more energy. So that's why there's a little qualifier there about the growth conditions. Can be aerobic or anaerobic, but it kind of depends on what type of organism you're talking about for fermentation. The final electron acceptor is always an organic molecule. Again, it's like we're not getting all the energy out of that organic molecule that the cell has taken in. And it can only carry out substrate level phosphorylation to produce the 2 ATP from glycolysis. Why do we want to study fermentation? Well, It is important not only from the foods that get produced, but it actually is very helpful in allowing us to determine the identity of many of our genus and species of bacteria. So in lab, we can do a simple fermentation test to see what sugars bacteria can utilize. This involves just taking a liquid broth and adding a specific carbohydrate. So let's just say, for example, something like lactose. We add in a pH indicator, phenol red. At a neutral pH, it's going to be red. And we also add what's referred to as a Durham tube. This is a little inverted glass tube that's going to be able to trap or collect any carbon dioxide that might be produced. So in tube A, we see the control. Nothing is growing, there's no turbidity, there's no cloudiness, and we see that the broth starts out red at neutral pH. In tube B, this organism is negative, so we see that the broth is still red and we don't have any CO2 being produced, so the organism is not able to ferment the carbohydrate, in this case, lactose. For tube C, we see that the tube has turned yellow. This indicates that acid has been produced, so the pH indicator is going to turn yellow when you have the production of those acidic end products, and therefore we know that the organism is carrying out fermentation of the carbohydrate and producing acid. Some organisms produce acid only, and some organisms can produce acid and gas. So in tube D, we see an organism that has produced acid and turned the broth yellow, but you can also see a bubble in the top of that Durham tube. That bubble is gas, and that gas is CO2. That again is a common end product of fermentation. So when we talk about the catabolism of carbohydrates, that's always the model that we use to talk about metabolism in general. But it doesn't mean that we can't use lipids and proteins to break those macromolecules down. Fortunately, though, lipid and protein catabolism involves the basic processes, the pathways that we already talked about, just different starting points. So some microorganisms can produce extracellular enzymes, meaning that these are secreted outside of the cell, and they will make lipases. These lipases break down larger fats or lipids into the byproducts or the building blocks of glycerol and fatty acids. So the glycerol is going to directly be converted into dihydroxyacetone phosphate, and that gets converted into our GP, our glycerol aldehyde 3-phosphate, and that's just the normal pathway of glycolysis. Turn that into pyruvic acid, converted in the intermediate step into acetyl-CoA, and makes the Krebs cycle go around. For the fatty acids, they are going to be oxidized by a process referred to as beta oxidation. You take off 2 carbons at a time until you eventually get that 2-carbon acetyl-CoA, and obviously that then is going to be shunted into the Krebs cycle to make lots of potential energy from Krebs. We also have protein catabolism. Proteins are broken down into the building blocks of amino acids, and this is again done by extracellular enzymes that are secreted, what we refer to as proteases or sometimes peptidases. Proteases are going to break down larger proteins, and peptidases break down smaller peptides, but they both produce the same building blocks, the amino acids that made the larger protein or peptide. Those amino acids can then be deaminated so that you take off an amino group, decarboxylated so that you take off a carboxyl group, or take off hydrogens, dehydrogenation, and they're converted into those intermediates which are referred to as organic acids in the Krebs cycle. So we can test to see if a bacteria can actually utilize a specific amino acid. So in this test, bacteria are first inoculated into glucose that has a pH indicator and a specific amino acid. We have to give them glucose to get started. It's the preferred energy source. Once it uses up all the glucose, then it starts looking for other things such as, oh, okay, can I use this amino acid? So in the first tube, we see that initially things turn acidic due to the fact that glucose fermentation has occurred and the tube turns yellow. However, if it can utilize that specific amino acid, then it's going to decarboxylate it, and you're going to have an alkaline reaction, and the tube is going to turn this purple color. So in that way, you can look for protein catabolism. So the beauty of protein catabolism and lipid catabolism is that they both just feed into the carbohydrate catabolism pathway that we previously studied, just at different points. So we see the lipids are going to be broken down into glycerol and fatty acids, go into glycolysis, and fatty acids make acetyl-CoA. The proteins get converted into amino acid building blocks that again can go into things like glycolysis proper, acetyl-CoA, and many of them are turned into the organic acids in the Krebs cycle. But they all are going to then feed into the electron transport chain, but it is the reason why when you have breakdown of proteins and lipids, you don't get as much bang for your buck as you do for that molecule of glucose. In reality, though, we've been talking about just the catabolic pathways. However, everything is really the amphibolic pathway, meaning that we have both catabolic and anabolic reactions occurring. So we break things down, but then the cell uses those building blocks for anabolic reactions to make macromolecules as needed. So simple sugars make larger polysaccharides, simple amino acids, the larger proteins for structure and metabolism enzymes. Glycerol and fatty acids get put into larger lipids and fats. And things like glycine, glutamine, and aspartic acid, which come off of the Krebs cycle, are then used to make purines and pyrimidines for things like DNA and RNA. So if we look at a couple examples, so some interesting things about the biosynthesis of polysaccharides, we see, for example, fructose 6-phosphate in glycolysis is converted into things like our N-acetylglucosamine and our N-acetylmuramic acid, our NAG and NAM, that is used as the disaccharides of the peptidoglycan, the cell wall of bacteria. For the biosynthesis of simple lipids, again, we see the glyceraldehyde 3-phosphate going to dihydroxyacetone phosphate. That can be converted into glycerol. Acetyl-CoA, the 2-carbon intermediate, can be linked together to make larger fatty acids. Combine the glycerol and fatty acids, and you get the simple lipids to make larger fats. The biosynthesis of amino acids, again, various organic acids in the Krebs cycle and things from the pentose phosphate pathway and the enterodeuterol pathway. These are going to go through a process of transamination where they basically are moving some of the components around to be able to make the amino acids. And lastly, we see the biosynthesis of our purines and pyrimidines. So again, glucose 6-phosphate is going to feed into the pentose phosphate pathway or the enterodeuterol pathway, depending on the organism, to give us our 5-carbon pentose sugars. And then things like glycine and glutamine and aspartic acid. These are going to come off of various points in metabolism to help synthesize our purine and pyrimidine nucleotides so we can make DNA and RNA molecules. So in reality, all of metabolism is integrated. We talk about it for the purpose of teaching the metabolic pathways, the catabolism as kind of this one-way street. But in reality, these are forward and reverse reactions so that for every time we have catabolic reactions, we usually have anabolic reactions as well. So again, you may have found the study of metabolism not your favorite thing, a little bit painful through the Krebs cycle. But in reality, biochemical testing is very important. And to do biochemical testing to identify bacteria, we need to understand their metabolism. So we use a lot of the metabolism to design biochemical tests to help us identify things down to the genus and species level. Many times we try to put these into what's referred to as a dichotomous key, where we can answer yes or no based on a certain biochemical test. So for example, let's say we did a gram stain and we know we have a gram-negative rod. So one of the first questions that we might want to ask is, can they ferment the sugar lactose? If no, then we go down the left-hand side. If yes, down the other side of the pathway. So let's follow down the no pathway, and we ask another question that we can look at of yes, no. Can they use citric acid as their sole carbon source? No. Well, it's probably a Shigella. Yes, it's probably a Salmonella. And we can do some supplemental tests, like see if they can produce hydrogen sulfide to verify things down to the species level. On the other side, if they can ferment lactose, then we can ask the question again, can they use citric acid as their sole carbon source? If no, then it's probably something like an E. coli. If yes, then we ask another question, can they produce acetoin from another metabolic reaction? And we see we can then differentiate the citrobacters from the enterobacters. So that's why metabolism is so important in doing our biochemical testing. From a clinical standpoint, we can look at an example for identifying Mycobacterium tuberculosis. So instead of doing a gram stain, we could do an acid-fast stain and identify something as an acid-fast mycobacteria. Out in the environment, there are what are referred to as rapid-growing mycobacteria. They can be problematic in wound infections and stuff. But some of the most problematic clinical ones, like Mycobacterium tuberculosis, is actually one of the slowest growing bacteria. So if we classify as slow-growing bacterium, we then can do something like a urease test to see if it has the enzyme urease to break down urea into ammonia and CO2. We can develop biochemical tests to look at when the ammonia is being produced, so again, incorporating pH indicators. So we can then differentiate and say, all right, can't produce urease? Well, it's probably Mycobacterium avium. It can produce urease? Well, we don't know, but we have to do another test. So we do something like nitrate reductase. See if it can reduce the nitrogen. If yes, then it's probably a Mycobacterium tuberculosis. If no, Mycobacterium bovis. So in this way, again, we're using the metabolism of the organism to help develop the biochemical testing that allows us to do our microbial identification. So in summary, we're going to try to put everything together and talk about our energy sources and classifying the ATP requirements for a diversity of organisms. As I said previously, we haven't had time, and we won't have time to talk about photosynthetic organisms. We're going to talk mostly about organisms that get their energy source from things like glucose, but there's a very vast microbial world that uses things like elemental sulfur and ammonia and various other things as their energy source. We focused mostly on the electron carriers of the NAD and FAD in their oxidized forms seen here. Again, those are in the catabolic reactions, and NADP is mostly involved in anabolic reactions. And we looked at the final electron acceptors. Aerobic respiration uses oxygen, anaerobic respiration, inorganics, things like nitrates and sulfates, and fermentation uses organic compounds as the final electron acceptor. So we can look at the nutritional classification of organisms and break them up according to those energy sources. If chemical, then we refer to them as chemotrophs. If light, then we refer to them as phototrophs. We can then further break them down based on their carbon source. So here, if we have a chemotroph, we can then look at what they use for their carbon source. If it's CO2, then it's referred to as a chemoautotroph, and these are going to be things like hydrogen sulfur and other weird oxidizing bacteria. If they're using organic compounds, then we refer to them as chemoheterotrophs, and we look then at the final electron acceptor. If oxygen, then that encompasses a lot of not only the microbial world and a lot of the clinical bacteria that we're going to talk about, but things like fungi and protozoa and all animals as well. If the final electron acceptor is not oxygen, then we can look at those things that are using organic compounds and carry out fermentation, such as Streptococcus. We can talk about those that are using inorganic compounds, so they're still going to be able to carry out respiration, but now it's going to be anaerobic cell respiration. So they still can use the electron transport chain, so things like Clostridium come to mind. For the phototrophs, same thing. Carbon source, if it's organic, then these are going to be photoheterotrophs. These are some of the environmental green non-sulfur and purple non-sulfur bacteria. If they utilize CO2, then these photoautotrophs are going to be classified as those that use water to reduce carbon dioxide. If yes, then these are the majority of our oxygenic photosynthetic bacteria and all the plants and algae. However, we do have organisms that can carry out what is referred to anoxygenic photosynthesis. These bacteria do not use water to reduce carbon dioxide, and they carry out a weird photosynthesis, but they don't give off oxygen. These are referred to as green and purple bacteria. So we can classify metabolic diversity by looking at things that are going to use light energy, which are going to be the photo, and things that are using chemical energy, which are going to be the chemos. We can then differentiate those by their carbon source. So photoautotrophs that use CO2 are going to be our photosynthetic organisms, both oxygenic and anoxygenic. We're going to see also some photoheterotrophs, unusual, but they are going to use light and organic compounds as their carbon source. And these are going to be some of the soil green and purple non-sulfur bacteria. Chemoautotrophs are going to use chemical energy source. CO2 is the carbon source. Very unusual iron oxidizing bacteria that sometimes we find out in like the runoffs from coal mines and things. But the majority of organisms are going to be classified as chemoheterotrophs. They use chemical as their energy source, and they use organic compounds as their carbon source. So most of the clinically relevant bacteria that we're going to talk about, they're going to be chemoheterotrophs, and indeed the protozoa and fungi that we are going to look at as some pathogenic organisms as well. So that concludes Part B. Again, make sure that you go through and look at the learning objectives for the chapter. We spent a lot of time talking about how we generate ATP from catabolism. We learned about oxidation reduction reactions and how that helps us to harvest our ATP. We talked about glycolysis and the pentose phosphate pathway, Krebs cycle, looked at the electron transport chain, all that under trying to figure out how we generate energy and how many ATP. We also took a little bit of a look at lipid and protein catabolism, but again, these feed into the regular pathway of our carbohydrate catabolism. And lastly, what happens during those preparatory stages for glycolysis? What are the principal products of Krebs cycle? How do we get all that ATP? What happens in aerobic versus anaerobic? And again, fermentation, what does it give us? Looking at the amphibolic, if we break it down, we use it to build it up, but we didn't spend much time on the anabolic processes, but be aware of some of the examples that we looked at.